bonjour à tout le monde. Un grand merci à Karim et Thibault et j'imagine il y a plusieurs d'autres personnes qui sont impliquées dans cet magnifique petit atelier scientifique. Est-ce qu'on peut utiliser le mot atelier pour un scientifique? OK, bon. Un grand merci, c'est toujours un plaisir d'être ici. OK, so this is really nice because I have the chance to feed off of Professor Coombs' lecture who spoke to us about hydroxyapatite. And I will spend most of my time talking about bones, whoops, sorry, pointer, about teeth and bones, okay, and a little bit about, at the end, the forgotten mineral that nobody thinks about. We do have calcium carbonate in our inner ears, and that's very interesting. So I'll end with a few unpublished slides about that work. Okay, so of course this is a slide that I use for groups that are not as expert as you are in biomineralization to illustrate, of course, the diversity of biomineralization. So I don't need to say anything about this slide to you. Many of you work on some of these structures, but most of you here also know that a hallmark of biomineralization, the naming of the biomineralization, is because there's this interaction with organic components. And I'm not sure, Dr. Garcia Ruiz, if I like the term biomorphs either, but we can discuss that with some wine, okay? So this is real bio, not Professor Garcia Ruiz's bio, okay? But they're all important, okay? All right, so I always like it. It's very humbling for me, and I like to step back from my biocentric perspective and integrate a bit of geology. I love this big canine tooth rock, and I use it to remind myself and to remind you that, of course, what has biology really done with geology? Well, biology has scaled it down to the nanoscale. So we have the same minerals, same mineral ions that we find on the crust of the Earth. You study those things. But the beauty of biology is that they can take it down to this level such that, as we heard from Professor Coombs, we have tiny nanocrystalline plates either within a collagen fibril, very, very tiny, or between collagen fibrils in bone, or we have these tremendously large, actually, as big as synthetic crystals of hydroxyapatite in tooth enamel, with every one of them extremely long, extremely similarly shaped, extremely identical dimensions. I mean, the biological reproducibility of biominerals is quite incredible, in my view. So this is an outline of my talk. I tried to squeeze in, I tried last night to think if I would have time to say something about matrix vesicles, and in the end I won't for lack of time, but if there's something important we can do it in the question period. But I'd like to talk about how these three aspects interrelate. I'll start the first part of my talk discussing with you some very interesting developments in the field of pyrophosphate and the enzyme that degrades in us pyrophosphate, which is tissue nonspecific alkaline phosphatase. So this is a very hot area, very exciting area. I've been involved in it, and I'd like to share that with you. And it's so exciting, it's even gone into clinical trials in patients, and I'd like to show you a bit of that. This uses many of the elements that I'll talk about from this topic and this topic, which is actual protein, peptide, amino acid interactions with a crystalline surface, in this case today, mostly hydroxyapatite. And matrix vesicle story is a bit of a combination of both. So if you understand, if I'm lucky enough to get you to understand those last two parts, you may understand a little bit about matrix vesicles. And then in the end, I have a very exciting opportunity to talk to you about a couple osteomalacia human diseases that I've been involved in. One, Catherine Chaussin is here, X-linked hypophosphatemia, and Paris has a very nice reference center for rare bone metabolic diseases. And I'll tell you a little bit about these two diseases once I've introduced the concepts that underlie those diseases and mineralization. Okay, again, this is useful for us. Let's step back. Let's remember we mineralize our skeletons. These are real human embryos, and you see the age of the embryos on top. This is real. The red is the hydroxyapatite or carbonatoapatite, substituted apatite anyways, that we heard about from Professor Coombs, beginning in a very young embryo, the red parts, okay, look at the nice mandibular development here, extending with time using cartilage which mineralizes, then using bone which mineralizes, and ultimately you get a mineralized skeleton. So you have a lot of mineralization going on in utero, of course, as you can see. Okay? And you need to get it at the right places at the right time. There's lots of calcium from the mother. Mother's eating well, not always eating at McDonald's, good sources of calcium, phosphate, and mineralizing hopefully that child will. All right? And then sometime along the way we also form these calcium carbonate autoconia. And so that's the go process. We build an extracellular matrix, and that's what's really great about bones and teeth. They're full of extracellular matrix, but your skin is also full of extracellular matrix. The dermis of your skin has the same collagen fibrils. So what's different about bones and teeth that lets them mineralize? Okay, so they all have an abundant, robust, cross-linked extracellular matrix for toughness. The mineral forms between the collagen fibrils, has a different morphology, the crystals, than within the collagen fibrils, within these very small hole zones or gap regions of the fibril, and there's a limited amount of growth takes place, and you heard the dimensions of these nanocrystals, they're very small. And then somewhat larger crystals form between these collagen fibrils over here. So we have two different biochemical environments, if you will, that we need to mineralize the bone. So this is good. This will mineralize, and this extracellular matrix will be completely impregnated with substituted apatitic mineral. And so here's a lower mag of that process, okay? So you have a blood supply, you have the cells that make the bone, you have the cells which make the bone, which are called the osteoblasts. You have a prefabricated collagenous-rich, but also non-collagenous proteins, extracellular matrix called the osteoid, OS. It's prefabricated, secreted by these cells, and then there's these foci of mineralization that start appearing, and then there's a massive escalation of mineralization coalescence at the frontière de la mineralisation, the mineralization frontier, and then you have mineralized bone matrix proper, here all in black in this TEM micrograph, and you embed and you trap some of these cells, get trapped and become embedded osteocytes, totally encased in that menacing bone, and that's really cool. I'll say a few words about that. So that's a good, normal, formative process. You want mineral. At some point, you have to stop mineralization, okay? If you don't stop mineralization, these osteocytes that just got embedded, here's an SEM micrograph, here's a TEM, similar things are happening in the dentin of the tooth. If you don't stop, completely inhibit with that nice healthy diet, full of calcium, full of phosphate, if you don't stop it, these cells would be obliterated. I mean, look at this menacing mineralization front encroaching upon this poor cell, sitting in its lacunae. Each and every cell is connected to its neighbor by these long cell processes, same in the tooth. The cell processes go in tiny little channels called canaliculi in the mineralized bone. These are only nanometers across. You can imagine how easy it would be to mineralize this little hole and destroy all these cell processes. But you need those cell processes to sense the mechanosensors in your bone. So you have to stop mineralization very carefully right there and right at the lacunar home of this osteocyte. All right, so we got a go condition and we have a stop condition for mineralization. So these are the players as we know today. Okay, there are probably others, of course. Nothing is so simple. Of course, we need our basic building blocks coming from a nice, healthy diet. We're not fish where we get calcium from the oceans. We are eating, periodically eating, and to obtain this, of course. Then there's a molecule, this is what I told you, has been well studied in recent years, called pyrophosphate, which is simply actually two phosphate groups. I'll tell you about that in a minute, stuck together. And so great progress has been made on handlers, genes and proteins that handle pyrophosphate in the body. And then this story is not just about phosphate, it's about organic matrix proteins, and I'll tell you in the last part of my talk a story about the phosphoproteins, and they're typically phosphoproteins, and they have this name of the sibling family, and they're really good mineral binders. And you know that if you have a mineral binder on a crystal surface, you can pin and stop growth steps, etc., terraces, and on crystals. So we need to talk about what binds to the crystals in our bones. And not only do we know a few of the molecular determinants, but actually they're processed by enzymes. They don't work necessarily in their intact state. There's a very careful degradation step by these green enzymes that you see, which will slowly modify these proteins, which are generally inhibitors of crystal growth. So you're going to have to remove them in a very careful way. It's counterintuitive that bones and teeth would have so many inhibitors of mineralization because they mineralize. It's like totally counterintuitive. But if you think that they need to be removed by enzymes that we know about, then you can allow mineralization of bones and teeth to carefully proceed to be regulated in a very controlled way. It must be. There are billions and trillions of crystals, and they all have to be the same crystal habit, shape, morphology, size, orientation, number, or else you will have some kind of brittle bone disease or soft bone disease, and I'll talk about a little bit about that. And of course you need a scaffold, a generalized scaffold, a collagen scaffold. So let's start our mineralization story on kind of the induction side of mineralization. In bones and teeth, actually, we know very little about nucleators. We know very little bit about amorphous calcium phosphates. We don't really know what nucleates mineralization. We know much more about once you have a crystal growing, how you can remove inhibitors and allow it to grow, to regulate it. Very little about nucleation. It's quite complex. So again, pyrophosphate is a molecule that's been known for a long, long time. It's in your toothpastes. It's used to prevent scale and mineral buildup in Paris's underground pipes. Basically, it is an inhibitor of apacitic crystal growth, and that's why it's in toothpaste and various places. There's the molecule, two phosphate groups with an oxygen bond in the middle. It binds beautifully to different types of minerals. The pharmaceutical industry has replaced the oxygen with a non-hydrolyzable carbon group and used this mineral binding property of a pyrophosphate analog to target osteoporosis drugs to the skeleton. Okay? There's a drug attached on this R group to this so-called bisphosphonate, and postmenopausal women mostly will take the drug, use this to target it to the mineral of the bones, and then the drug will start killing the resorptive cells. All right, so it's a targeting motif, and so it's been used around for a long time in the 60s from these guys, and it's used for many purposes, but the real processing and handling of pyrophosphate has only come around in very recent years. And it binds to calcium, and it can bind to crystal growth surfaces, as you see here. All right? And so there is an enzyme. Okay, so bones and teeth have all these inhibitors, but you want bones and teeth to mineralize. So this is an inhibitor. It's systemic. It's floating around in your blood. It's in all your tissue fluids. It's in your skin. It's a systemic inhibitor of mineralization. It turns out that bone and tooth cells make abundant amounts, and that's why this works, of an enzyme called tissue nonspecific alkaline phosphatase, TNAP or ALPL. And it's precisely because the cells in the bones and teeth make this enzyme that can cleave and remove this inhibitor in bones and teeth, thus allowing mineralization to get started. And so the enzyme will cleave inhibitory pyrophosphate. In so doing, generate 2 phosphate ions that in principle could even be used for crystal growth. Okay? So it's a kind of 2 birds with 1 stone expression. You remove an inhibitor, and you actually also generate phosphate, and you can mineralize your bones and teeth. So that's a key message, is that mineralized cells, mineralized tissue cells, make abundant alkaline phosphatase. Okay? That's why the skin doesn't mineralize. You can put alkaline phosphatase in the collagen scaffold of your skin. I think I have the slide coming. And you can mineralize your skin. I'll show you. All right. Just to show you the potency of pyrophosphate, this is a cell culture system we use. It's a mouse bone cell culture. It grows nice, thick sheets of bone, in a sense. You can actually peel it off the dish like a plastic sandwich wrap. It's that robust. The black is mineral. The light blue is extracellular matrix. The dark blue are the cells. This black is the mineral. So under normal conditions, you can get nice black mineral, lots of calcium, mineralizing. You toss in a little bit of pyrophosphate, you can completely block that black mineral from precipitating. It's a very potent mineralization inhibitor. Okay, it binds to crystals. You simply toss in a little bit of recombinant enzyme that the group in Montreal has made. It can cleave away that pyrophosphate. You're putting both in, and you rescue the mineralization. Okay, just a very simple demonstration of the efficacy of pyrophosphate as an inhibitor and its degradation by an enzyme to allow mineralization to proceed. So these are the exciting recent experiments. In recent years, a number of mouse knockout models have been made where the enzymes that process pyrophosphate and transport it across membranes, one called ANC. Pyrophosphate is everywhere, remember. There are transporters that get it out across a cell. There are nucleotides in the extracellular matrix that you can make pyrophosphate from. These are the enzymes and transporters that do it. One by one, they've been knocked out in mice. So if you remove two molecules that produce pyrophosphate, okay, so what happens there? You have no pyrophosphate or less pyrophosphate, okay, and you start to have over-mineralization. Joints start fusing, bones become more mineralized. If you, on the other hand, you remove the enzyme that cleaves away the inhibitor, okay, you have too much inhibitor accumulating out there, and mineralization cannot proceed because there's large pools of pyrophosphate blocking the crystals. So all these three, four mouse models made sense, and the story was actually quite surprisingly clean. Yeah, here's the other bit I was telling you about. You can use a collagen gene promoter that's highly expressed in the skin of a mouse. In other words, I told you there's collagen in your skin, there's pyrophosphate everywhere in your body, blocking mineralization of that collagen. If you artificially put this enzyme in a transgenic mouse, you can mineralize the skin. This is the epidermis. This is the surface of an arm. In this case, it's a tail. Here's the epithelial cells, of course. There's no matrix. Here's the dermis of the skin. Here's hair follicles. It's mineralizing. And these sheets of mineral around the tail vertebrae of a mouse show you very clearly you're building this exoskeleton, if you like, in the dermis of this poor mouse. It's a crunchy mouse. And if you go in with electron microscopy, you cannot actually distinguish whether this is a piece of bone collagen mineralizing. In fact, this is a piece of skin collagen mineralizing with these nanocrystals. So you can mineralize tissues that you don't want to mineralize. So the story continues, of course, because there are many, many mutations, more than 100 in humans, in the alkaline phosphatase gene, the TNAP or ALPL gene. And depending where those mutations are in the gene, you will get different severities of enzyme activity, from mild to strong, okay? You mutate an enzyme, you can change its catalytic pocket, and it might not be able to degrade pyrophosphate. And so that's what happens. Patients with TNAP mutations have soft, rachitic type bones, not nutritional rickets, but they have soft bones that have these kinds of pseudo-fractures. They bend and they deform. And here is and we call that osteomalacia, when all this bone matrix is produced, and actually the bone cells are fine. They're making lots of bone matrix, all that blue. There are some of the cells, but only breakthrough spots of mineralization. It's hypo-mineralized, and thus the bones are soft. Okay? And the teeth in these patients also fall out. Now, what's wrong with these teeth that fell out? Those of you who had children, or those of you who have good memories when you were under 5 years old. There's something really wrong here. These teeth have roots. You know that just the crown attaches to the gum. You wiggle, you tie your string on the doorknob, you slam the door, out pops the crown, right? No, these have roots. Something's really wrong. They're just sliding out of the bone. What's that story about? I'll tell you a little bit about it. So there are some people in this room that know very well there are different types of hypophosphatasia depending on the stage. And the most severe forms are the earlier forms, children born at birth, and when they're young infants. Actually, it's much greater than 50%. The majority of the kids that have certain mutations in alkaline phosphatase die by 1 year of age. And the greatest population of people with this disease is in Canada. Did you know that, Catherine? Yes, in the Mennonite population out in Western Canada, Midwestern Canada. It's a rare disease, 1 in 100,000 live births, but in Canada we have a group that has very frequently hypophosphatasia. And their kids, they lose 1 or 2 kids a year born with this disease and die. All right. So a while back, there was a Montreal, this is kind of a success story, and I'm very proud of it, because as a basic scientist, it's very rare to actually work with a company and develop a clinical treatment. And I'll show you some kids that got treated with this treatment. So to be involved in that has been really rewarding to me. And France has a very nice national reference center network for rare bone disease patients, and some of the people in the room are involved in that, so it's nice to be here. So knowing that these kids don't have, and adults, don't have functioning TNAP enzyme, they have mutations, there's something wrong in their protein, all right? A company in Montreal, a biotech startup company, developed a recombinant enzyme replacement therapy. Let's give back these kids some TNAP. Let's inject it into them. But wait a minute, if you inject it under the skin, you might mineralize the skin. And if you inject it in blood vessels, you need to get it to the bones. It's going to go everywhere. So they developed, and I worked with them on this, they developed a bone targeting strategy. Here's the enzyme, here's some spacer stuff, and that red part is the important part. Instead of using a bisphosphonate like Big Pharma does for osteoporosis drugs, they used a peptide sequence, a negatively charged polyasp deca 10 amino acids that we know bind to calcium because they're in the proteins that I'm going to show you, and they allows you the possibility to make fusion proteins recombinantly with a targeting motif. Instead of pyrophosphate analog, you actually use a peptide sequence, and I'll show you why that works, to target this enzyme to the little skeletons that those kids had, the little mineral, they have some mineral left in their bones and teeth. And so this was a strategy to send TNAP to the bones. And so the reason that I and others were working with this company, I've been studying almost my entire career a protein called osteopontin. It has this polyasp mineral binding sequence. It has others, and I'll say more about it later. But we know how some peptide motifs bind to hydroxyapatite mineral. So the logic was there. It made sense. We know that the polyasp region of many proteins, acidic, negatively charged proteins, will bind to the calcium in hydroxyapatite crystals. Well, lo and behold, it worked. You inject it in the preclinical trials in the mice, and there were knockout mice and everything. You inject it, and it goes to the bones. That polyasp sequence, we modeled it using Rosetta Surface. We found which carboxylates bind and coordinate with calcium atoms on the surface of, this is hydroxyapatite. The carboxylate side chains of the polyasp sequence dock onto the surface. And so when you inject a kid with it, that pyrophosphate here binds to, by its red polyasp tags, binds to that little bit of mineral that they have. You've docked an enzyme onto that mineral, and this enzyme is a pretty tough mineral, and it starts to work. It starts to work by removing all this pyrophosphate that's flooding this area and stopping this bone from mineralizing. The enzyme starts chopping away, and the bone actually resolves and mineralizes properly. And these were preclinical trials in mice that were done. And here's here's a mice. I just have one slide, of course, this whole studies, but just to drive the method. Without alkaline phosphatase, these mice would die by a couple weeks. Okay, these are the percent survival curves. When injected with the drug, the TNAP, here's the survival course here. Okay, 75, 80% of the mice were now living up to 2 months. So it saved their lives, these little mice. When you look at a mouse, this one here, that does not have alkaline phosphatase, the same as these kids, these mice look like this. This is a section through the bone of the mandible of a mouse. There's the incisor. There's the surrounding mandibular bone. They've got pretty good black, but look, huge patches, the red asterisk, of unmineralized bone, osteomalacia. Huge patches of unmineralized dentin of the tooth. It should look like this. It should look like this, but the black is missing. But the tooth structure is there. The bone structure is there. It's just waiting. This is the beauty of it. It's just waiting to receive its mineral, but it can't because it's flooded with pyrophosphate because there's mutations in the enzyme that can't get rid of it. So when you give, in the green box here, when you give the drug, sure enough, you can really nicely have mineralized bones all the way around the tooth, mineralized tooth. Here's molars, no mineralization. You give the drug, mineralized roots, just like in a wild-type normal mouse. So in preclinical trials, it really did work well. I don't know, the dentists in this room would understand it, but the reason that these teeth fall out with their whole roots is because they are not attaching well via the periodontal ligament in the jaw bones. And they slide out because they're not attached. They're attached because certain proteins are supposed to reside on the surface of the tooth to attach them to a ligament. And those proteins are not there in the knockout mouse. Okay? There's not this red layer of a protein called osteopontin. There's not a tissue called cementum to attach the tooth to the bone, and these teeth just fall out. When you give the drug, actually this layer of acellular cementum is reestablished just like a normal mouse at the root surface. And we haven't done the studies yet, but it seems that their teeth would be much, much better. So I'm going to show you a few patients because this has really been rewarding for me. Based on the success of the clinical trials, the mouse survival rates, this went quite quickly into some clinical trials. It was started in Montreal, and then it was acquired by a larger company in the United States where the drug's expected to be on the market this year, 2015. In Canada, maybe 2 years later for that Mennonite population. But these are the kids that are born at birth, and they can die immediately, or they're born already dead and they have broken bones and everything from the delivery because they have soft bones. And these are the kids that survive a little bit better on your right side of the screen, but they're going to die by about 1 year of age. They failure to thrive. They can't breathe properly because their ribs are too soft. They have kidney stones. Their bones are fracturing. They're having seizures. They're really in bad shape. So the company called Inobia, which you see in the corner back then, started its clinical trials with this mineral targeting drug. And this was the first little girl that was treated, Amy from Belfast. She came to Canada. And she was treated by a physician who I just met last week, actually, in my office. The clinical trial had originally 20. Now it's 25 infants, and older kids are now being treated. And Amy was going to die very soon. They had weeks to live was the prognosis, and she went right away on this clinical trial. And she was a pretty sick baby, but she did very well. And this was the state of her bones when she came. Okay, this is not, for those of you who know, this is not how a bone's supposed to look. This is how it's supposed to look. It's all under-mineralized. You know, there's lots of pyrophosphate floating around in this knee. Her hand, her finger bones were not even developed at all. There was matrix there in the fingers, but it wasn't mineralized. She just had these little bits of soft mineralization in the palms of her hand. And here, after treatment with 1 year, getting injections in the skin 3 times a week with this mineral targeting enzyme, this is what she looked like 21 months after 1 year. 1 year after treatment, and here after 1 year treatment. That's not, yeah, there's a little curvature and deformities and bending, but that's not too bad. Oh, that's another patient. And then she lived past a year, and she grew older. And these are her very first steps, captured on film. And from her treating physician who visited me last week, this is a little bit later. She's starting to run, play soccer. Yes, I guess. And then here she is blowing kisses at somebody. So des bisous, all right? And there's the doctor, Dr. Greenberg, who treated the very first patient with this drug. So there are 25 kids been treated. They've all surviving. Only one died from pneumonia. That was an unrelated infection. And the Food and Drug Administration in the United States gave it breakthrough status last year to accelerate its development. All right, so now I'd like to talk in the next part of my talk about this idea, about peptides, amino acids, proteins binding to crystal surfaces. It had implications for origins of life. It has implications for diseases, as you saw, human diseases. What do we know about, and my focus will be on bone proteins. What do we know about bone proteins in vivo, in the real environment, that bind to the surfaces of crystals? What I've shown you really is about small molecule pyrophosphate and an enzyme. Let's talk now about bigger molecules, okay? And you know mineralization is happening in a very complex soup of proteins and degraded peptides from those proteins. The idea that proteins could control mineralization in vivo was not shown until this study. Okay? And this was not even, I'll tell you about that in a minute. Everybody had been talking, they've been doing in vitro crystal growth assays, gel growth assays, solution growth assays, and it all made sense using the proteins, but there was no genetic, definitive animal in vivo evidence that proteins could really do that in the physiological setting. So working with Gerard Carcenti at the time, way back, worked on this one particular protein, which exactly is not really a bone protein. We weren't sure where it was at that time. It turns out it's a blood vessel protein and a cartilage protein. And when you knocked out this protein in 1997, the entire vascular tree calcified in these poor mice. Okay, here you go. You know, this was quite remarkable, and it was 100% penetrant phenotype. In other words, every mouse developed this and died by 1 month of age. They died because they were happily playing rambunctiously in their cage, and their mineralized aorta snapped and they hemorrhaged to death. Okay, so their blood vessels in every single organ and tissue started to mineralize. The blood vessel extracellular matrix, not the bones. The bones were fine. The blood vessels. The collagen in the blood vessels started to mineralize. It started in the aortic arch, descended like a wave down the aorta, and spread out towards all the blood vessels until it was too much and the mice died. Here you can see where the black mineralization occurring. It occurring in the wall of the blood vessel. This is a normal mouse aorta. This is a calcified, sheets and sheets of mineralization through the elastic lamellae, through the collagen, and basically turning that into a stiff tube. The electron microscopy, you can see the mineralization started in the elastic fiber lamellae, went down as waves, and then extended out into the collagen on each and every side, depending on how long the mouse lived. So that was the first genetic evidence that a protein could control mineralization of extracellular matrices. Again, with Gerard Carcenti's group at the time, we said, well, it's not really, it's in blood vessels, it's not in bone. What would happen if we flooded bones and teeth with that protein? So that's what we did. We used specific promoter fragments to drive that protein, MGP, matrix GLA protein. And look what happens when you overexpress it in a tooth. There's the molar. Look, it's got its roots, just like pyrophosphate situation, but they're not mineralized. A few breakthrough mineralizations. Look here, the incisor of the mouse. It's supposed to look thick and mineralized like this. Completely half of the tooth is not mineralized, and these teeth were just breaking off. The tooth forms, but it doesn't mineralize. Okay, and here's some three-dimensional pictures of that. Okay, so we had proof, but now we needed to know, and we artificially made the protein in bones and teeth, but what about the real bones and teeth proteins. So the idea is that the real bones and teeth proteins, between the collagen fibrils where there's room for them, can control crystal growth to give you, depending on which faces of a crystal that you bind something to, a crystal poison, you can shape and size the crystal. So you can get these various types of crystals. The idea here is that proteins dock either by themselves, with binding partners, single layer stratified, some might even get occluded inside the crystal structure somehow, nanoparticles or lattice structure. These are the questions in the field. But certainly some kind of binding event would have to regulate and shape crystals that could be so different from tooth enamel hydroxyapatite to bone hydroxyapatite. And the idea, just like the pyrophosphate, is that peptides or proteins could come onto the crystal growth surfaces and somehow guide the way a crystal might go. And this is important. It's not trivial. Anything wrong with shape, size, number, orientation, location will give you a bone or tooth disease. All right. So it's been known that besides the pyrophosphate story, there's a family of proteins, I'll explain this in a minute, out here in the matrix that can bind to the hydroxyapatite and regulate the growth. Not only that, it turns out that pyrophosphate and phosphate are signaling molecules that can come through channels and actually change the levels of these mineral binding proteins. So we have a loop, a phosphate, pyrophosphate protein loop. There's also calcium loops. I won't talk about them. So let's go now. We already talked a bit about these small molecules. Let's go out into the matrix and look at the features of these mineral binding proteins in bones. It turns out there's a family of 5 proteins. Some of the mutations and some of the diseases, dentinogenesis imperfecta, rickets, etc., are known for some of these. Others are not known. It's a gene cluster that evolved from gene duplication from spark or a spark-like ancestor over 300 million years ago. Highly conserved family. They're part of a larger calcium-binding family called the SCPP, phosphoprotein family, and they're phosphoproteins, not cytosolic signaling cascade phosphoproteins that have 1 or 2 phosphorylation sites. These proteins have lots of phosphate groups on them. Why is that important? Because phosphate can bind to calcium. So in all the calcium-based minerals, these proteins can bind. They can bind to calcite, and they're found in eggshell. They can bind to autoconia, and they're found in autoconia. They can bind to bone and tooth crystals. All right? They're very flexible proteins. They have lots of phosphorylations, and they're very negatively charged. 25% of the amino acids are acidic residues like aspartic acid and glutamic acid. Oh yeah, so this just takes 2 of those proteins. And this is the neutral pH right here. In the phosphorylated state, look how negatively charged they are. Osteopontin in phosphorylated state has a pI of 3.4 acidity for this protein. A little bit higher for MCKEE, and higher when it's phosphorylated. So just look at this one. Look below the neutrality point. Look down here. And so people in the field, such as myself, have focused on the most acidic region, in this case osteopontin, a very, very negatively charged sequence called the ACE-arm peptide, and I'll talk to you about that. There's also the polyasp that we used for the drug. That's another part of these proteins. But just to show you how negatively charged they are and how easy they could bind to positively charged lattice calcium, for example. And not only that, they share this motif. The ACE-arm motif, these are the sibling proteins, some of them. The ACE-arm motif is shared, duplicated in all of these proteins. And here it is. This is the osteopontin sequence. Look at all these acidic aspartic acids, glutamic acids, all in a stretch of about 18 mer stretch, 5 potential phosphorylations along this site. So this is something we've studied because we can make the peptides, we can model the peptides. It's a little bit harder to work with the whole protein. So that's that very acidic region coming back to this side. Here's another one next to it called the polyasp. It's important to note that the phosphates are distributed throughout the there's clusters, but they're more or less distributed along the entire length of the sibling proteins. That's important. It means that these proteins can be extended and bind to many calciums across a larger, infinite surfaces, all right? So the phosphorylations are important. The proteins can be cross-linked covalently into large polymer networks. That's another story. And they're processed by enzymes. Not only that, the sibling proteins like osteopontin, that's the best studied one, are extremely disordered, loose, floppy proteins. In fact, it was thought that transcription factors were the most intrinsically disordered proteins, but it turns out, publication 1 or 2 years ago, shows that the sibling proteins are now officially the most extended, loose, floppy proteins that there are. Perhaps to go and find calcium atoms and cover a larger surface of a growing crystal. All right? 25% acidic amino acids, that's a lot. Clusters, okay, and very disordered, and it can be processed by enzymes, and I'll show you what that means. This is a recent paper from Esben Sorensen and colleagues, just showing that they mapped using titration calorimetry, they mapped where the calcium binding sites are in solution. This is not about a crystal surface, but they think that these calcium atoms, 10 or so, very strong chelation binding, can be replaced by lattice calcium as one of these proteins unzips and flops down onto a crystal surface. I have some movies of that. Okay, so this is just a pretty nice paper that just came out for the solution state of osteopontin. Proof that the proteins bind to crystals. There are many ways to do it, but here we use, and it's a common theme in my work, I do a lot of immunogold particle labeling with antibodies to know where these proteins are on crystal surfaces. You can make synthetic apatite, throw in slurries of osteopontin, and sure enough, it binds very well, and it binds better when it's phosphorylated, of course, and you can see all the gold particles. What about real bone? That was synthetic apatite. Real bone, these are these very first mineralization foci that I showed you, where there are thousands of nanocrystals of apatite forming, and sure enough, osteopontin in this case is very prominent in each and every one of these initial mineralization foci, we call them. So it's there where it should be if it's a regulator. Remember earlier I mentioned that sometimes you really have to completely block mineralization so the osteocyte can survive and those delicate cell processes can survive. If you look at those two sites, you have very high concentration of osteopontin as shown by the gold particles lining the edges of where you want to stop mineralization. So that's good evidence that osteopontin also is binding and is a potent mineralization inhibitor on the walls of the bone. Just like pyrophosphate, we did the same thing. You toss in a little bit of phosphorylated osteopontin and you can completely shut down, like pyrophosphate did, the mineralization. You add, oh yeah, by the way, TNAP, that same enzyme I talked to, is a phosphatase and can dephosphorylate some of these sibling proteins. So remember, the cells are making also TNAP, and so you can modify binding and phosphorylation also, another function of TNAP. And when you toss in some TNAP to strip off the phosphates, you can rescue a lot of the mineralization. Okay, so that's an in vitro demonstration of what's going on. I thought this was interesting just to come back to that mouse model where the blood vessels were calcified here. So these mice are calcifying and dying their blood vessels because they don't have MGP. It's a knockout mouse. All right. We went to look at if they don't have their main inhibitor, MGP, what are they doing to try and fight this terrible mineralization of their blood vessels? And we thought that they might be using osteopontin, which is also a potent inhibitor, upregulating their next best thing. And sure enough, we found by immunolabeling lots of osteopontin on the surface of the calcified lamellae. So they're trying to fight the mineralization to live, okay, with the second best inhibitor for blood vessels. They decided to use osteopontin to do that. It was the right place. So working with CCG Akeli, made and analyzed the double knockout mouse. They don't have MGP and they don't have osteopontin to fight that mineralization. And sure enough, those mice deposited much more mineral in their blood vessels and they died 2 weeks earlier than the MGP knockout average lifespan. Okay, so one by one you can start taking away inhibitors and make these pathological calcification situations worse. These are not just mice. This is from a heart transplant patient. This is a coronary artery of a guy who had bypass surgery. There's his coronaries. Here's the wall of his blood vessel. He has atherosclerosis. Here's a massive hydroxyapatite mineral or apatite mineral. Here's the blood in the lumen blocking up the wall. And when you go and look in osteopontin in human coronary arteries, you're finding tons of osteopontin on all those mineralization foci. Okay, so it's not just mouse, it's humans. And Willi Jan Deschant in Germany, worked with him for many years. He's knocked out another inhibitor that's made systemically by the liver called fetuin or alpha-2-HS glycoprotein. And certain strains of mice, if you remove that circulating blood protein called fetuin, almost every tissue in the body can mineralize. You can't knock out pyrophosphate, it's a small molecule, but you can knock out protein inhibitors and genes. And this situation here looks really uncomfortable for me, but every tissue was mineralizing. And when you go and you look in those mineralized tissues, here are others, pancreas, lung. There's the black pathologic mineral deposits, and immunohistochemistry for osteopontin here in red shows that the local cells are massively upregulating osteopontin to try and inhibit this pathological calcification. Kidney stones. I was supposed to talk a little bit about pathology, so I've just thrown in a few slides about kidney stones. Okay, do kidney cells, well actually this was kidney cells. No, this is not kidney from that mouse model. Kidneys, there are models to make kidney stones in mice. Okay. One kind of mineral that's prevalent in kidney stones is calcium oxalate. This happens to be calcium oxalate dihydrate. Okay, so you know kidney stones are mineral. Is osteopontin upregulated in kidney stones? So I'm going to show you two types of kidney stones, calcium oxalate dihydrate and apatite stones. In this case, you feed the rats. This is rats. You feed them ethylene glycol. They get these kidney stones. This is what they look like in scanning electron microscopy, unprocessed, these dumbbell-shaped. Here's a fully demineralized specimen to show you how much protein. How much protein. These are delicate shells, but this is all protein, no more mineral. Mineral and protein, no more mineral. And when you zoom in at the crystal surface, you see individual tablets of calcium oxalate. You see this lumpy, bumpy stuff at the surface. What is that? Well, that's a protein coating. If you cut it and section it and do some immunolabeling, in this case for osteopontin, you see that the calcium oxalate is covered by a coating of, we're guessing, inhibitory osteopontin. The cells, these cells nearby, right here, the epithelial cells in the kidney, are producing from the Golgi apparatus, secretory granules, secreting and releasing huge amounts of osteopontin that bind and accumulate in the nuclei of these dumbbell-shaped calcium oxalate crystals, and then kind of like growth rings on a tree, and the onions and paleobiology and coral skeleton cores from divers, they grow sequentially. You know, you inhibit, but then they grow a little bit more, and then you inhibit them again. Is a regular feature of human kidney stones, are concentric growth rings. Each and every time, though, there seems to be an attempt by osteopontin to limit that mineralization. So we wanted to study a bit more the interactions of some of our molecules that we're interested in, motifs in osteopontin, let's say the polyasp motif that I've already talked to you about. We grew our own calcium oxalate crystals, which normally would look like this. When you toss in a little bit of polyasp peptide from the osteopontin structure, you can bind to certain faces, like I discussed with you. You can elongate some and not others. And the crystals, instead of looking like this, they start to look like this. Not only do they bind to the surface, but at low doses, you can actually see that they're incorporated by sectoral zoning. Whoops. They're incorporated by sectoral zoning into the individual crystals. You see there's more fluorescent. You can use a fluorescent marker on the peptide, and you can study how peptides are incorporated into a calcium oxalate crystal, peptide occlusion. It's very important and very interesting. And we modeled using Rosetta surface, we modeled how we think the polyasp binds to the calcium in the calcium oxalate 110 surface. Using our other peptide that we like from osteopontin, we grew crystals. We could remarkably go from a normal crystal to very similar that we saw in vivo, dumbbell-shaped crystals, lots of calcium oxalates. They extended and grew in various ways, and this is now with a completely different peptide motif. So the two peptide motifs do things a little bit differently to these calcium oxalate crystals. But it's remarkable how they actually reproduce that dumbbell. I don't know if it's a twinning kind of growth or what. We're preparing a paper on that now, and I can speak to Dr. Garcia Ruiz about that. All right, and then the calcium phosphate kidney stone model. You can put kidney stones in a mouse by making the pee, the urine, high in phosphate concentrations, right? So you have renal phosphate wasting, and you have lots of phosphate. This is common in bone metabolic diseases. You have lots of phosphate in your urine. So you start precipitating hydroxyapatite stones in your kidneys, and that's what we have here. Osteopontin, as before, is upregulated in each one of these stones, here shown in red, here by immunogold labeling. It is hydroxyapatite mineral. So it's another example. It doesn't seem to matter what kind of mineral you have. The body will fight it with the inhibitors it has at its disposal. All right, so coming back now to this ASARM motif. It's important because patients have elevated levels of this ASARM motif. Okay, so there it is. I've already showed you this slide. All right, we made this peptide. We synthesized it with various phosphorylation levels. So the reds are with 3 phosphate groups. Here, 3, none, 1, 2, 3, 4, 5 phosphate groups in that ASARM peptide. Okay, so we did use as experimental tools to see the importance of the phosphorylation. Again, we actually made motifs from 2 of the sibling proteins. This is now those peptides. The phosphorylated one, the P, binds much better to the crystal surfaces, the apatite, than the non-phosphorylated forms. You can measure it this way. You can measure it in depletion from supernatant. There are ways to measure it. So the ASARM peptides were binding well to the mineral when they're phosphorylated. You could toss them into the cultures, just like we did for osteopontin, and the phosphorylated ones, P in particular, would bind to those early mineralization foci. So it was going to the right place and where you would expect it would go. And the number of phosphates, 5, 3, and 0, and a neutral, would dose-dependently, here's the black, here it's a bit orange, dose-dependently inhibited mineralization of our bone cell cultures. Okay, so ASARM was working like pyrophosphate, like full-length osteopontin, like polyasp used in the drug treatment, and ASARM was doing very similar things. And importantly, ASARM is found in patients. Here's a modeling sequence of 3 slides showing the Rosetta surface modeling technique. And it's really a nice, powerful technique. It's been validated by solid-state NMR. It was originally used for David Baker's group in Washington for protein-protein docking, antibody, ligands, etc. Jeffrey Gray at Johns Hopkins, modified it for mineral. And what he did was he was able to dock in low energy, this is energy minimization, supercomputing peptide low energies, okay, was able to throw in a constructed slab of mineral. And it's a very nice, powerful tool, validated again, as I told you. And you build and you find your low energy states, and you actually sample 3.5 billion conformers by supercomputing over a span of a few days of a peptide this size, 18 mer. And you find how this peptide docks and finds its low energy states on a constructed slab of hydroxyapatite. And Professor Coombs, I know we have to pay more attention to the hydrated state and the carbonate substitutions, you know, and the HPO4 groups. But we started with hydroxyapatite and got some really good binding energies. You take the top 100 energy minimization conformers of that after sampling 3.5, and you get the binding energies in Rosetta surface units. And the phosphorylated, the highly phosphorylated peptides were the best binders compared to 3 phosphates, 0 phosphates. Here's the binding energies, okay, and they often extended out along the surface lattice calciums like that. So that's what the modeling showed us, and it fit very nicely with the in vitro data. And from those top 100 scoring models, you can actually go in and find out which amino acids in your peptide were closest to the mineral surface, and it was always the phosphates and the carboxylate groups in these very acidic peptides which were coordinating with surface lattice calcium. All right, So Catherine Chaussin and others in this room have noticed that in the blood of patients that they treat with soft skeletons, that ASARM is elevated in a disease called X-linked hypophosphatemia. They have high circulating amounts of inhibitory protein. And they have soft skeletons. Maybe it's inhibiting their skeletons via the circulation. Okay. And so mutations causing this disease are found in an enzyme called FEX. Nobody knows what the substrates at the time were for FEX. People have been searching for 15 years what the substrates were for FEX. And this is not as lethal as hypophosphatasia, but the patients have very soft, also deformed skeletons, osteomalacia. We knew the enzyme with the mutations, but didn't know the substrate. So we decided to look if that ASARM peptide, which was elevated and may be contributing, in addition to renal phosphate wasting, to the osteomalacia, could the ASARM be a substrate for the known mutation enzyme? Okay. Sorry, I'm jumping the gun. That's the next slide. This is just to show you the patients, the osteomalacia, like in hypophosphatasia. And this is from Catherine. This is the teeth, actually, are hypomineralized also. This should be all blue in here, all blue mineralized. It's got what I call dentinomalacia. Okay, so soft teeth, they get infections. Soft bones, they get deformed bones. All right, let's skip that. And the enzymes are made in the bone. This is from Catherine Chaussin's group, where she identified elevated MEPI-ASARM in patient teeth. Because she works on that and because it's a national reference center, she accesses that and treats those patients. There's a tooth from one of the patients. All of this should be black. It's not. It's soft. And the patients have, by mass spec and by gels, they have the red arrows, they have increased levels of this ASARM peptide compared to control patients, which are not underlined here in red. All right, so there's a disease just like we did. So what happens if we drop in our ASARM peptide in our culture system? It blocks mineralization. What if this was our first evidence for the new substrate. We tossed in some FEX, the green arrow, and we could rescue mineralization. We could degrade, it seemed, the inhibitor for both the MEPI-ASARM peptide and the osteopontin peptide. And with mass spec, we were able to notice a complete degradation of, complete degradation from here to here. This is how you show substrates by mass spec. FEX was degrading MEPI-ASARM, and we were able to map the exact cleavage sites within that ASARM peptide. So this fit with the XLH story in the patients. Yes, and we also mapped the cleavage sites for the osteopontin peptide, ASARM. Remember, all those proteins have one of these acidic stretches. And it was the same thing. So we identified a substrate. It was not a whole native protein, but we identified the first substrate for that enzyme with those patients. So the idea is, the working model there was that the ASARM peptide, would normally, in you and me, would normally be degraded away. It's inhibitory, but it's degraded, and you get inactive fragments, and mineralization can proceed. In the disease states, there's no FEX to degrade these inhibitory peptides. They accumulate in the bone, and you have a red light, you have osteomalacia. But we know that if you toss in osteopontin, the full-length protein, it also blocks mineralization, and just like the peptide. So could full-length osteopontin be a substrate protein, parent substrate protein for FEX? So we checked it out. And I'll go quickly through this because I'm running out of time, but sure enough, this is full-length osteopontin. You add some FEX with time, you degrade it. This is how you show degradation studies. New bands accumulate lower, degrade it with FEX. New bands go lower. You can add inhibitors. And finally, by 4 hours, full native length OPN was cleaved into these smaller fragments. So we were able to identify not just its peptides, but the full-length osteopontin protein. And here's the shift. We don't have to go in detail, but here's parent protein. Here's the shift with time over 4 hours. No more parent protein, larger fragments, very small fragments. This is how you demonstrate a substrate for a protein. So we had an enzyme that could degrade one of our most potent known inhibitors in extracellular matrix biology, degrade it. And not only degrade it in one place, like enzymes usually do, degrade it to dozens and dozens of tiny little peptides. And in fact, the potential cleavage sites are all over this molecule. The first one, actually, cleavage site occurs in the ACE arm peptide, but they're everywhere. These little arrows are all potential cleavage sites based on the substrate specificity requirements for the enzyme. So this enzyme was exhaustively destroying a mineralization inhibitor. Okay? And the patients that don't have this enzyme were not destroying their osteopontin, and it was accumulating in their bones in addition to renal phosphate wasting. So we checked that out. Okay, I'll skip that. We checked that out. There's a mouse model and there's human patient samples. This is brown, is immunostaining for osteopontin at the light microscope level. These dots are the gold particle immunolabeling. Look at the bone. Look at all the black dots in the bone of the mouse model of XLH compared to normal bone. Tons and tons of osteopontin accumulating in the absence of effects. We looked at human bones just recently. This is unpublished. This is a normal piece of human bone. Here's the trapped osteocytes. A little bit of osteopontin. Here are the XLH patients. Huge amounts of osteopontin accumulating around the cells for the most part, but accumulating in many places. So in humans too, it made sense. So it's not just an ACE arm peptide story. It's an osteopontin story and its peptides and other peptides that are contributing in some of these diseases to the osteomalacia. In some cases, also with renal phosphate wasting. But the FGF23 knockout mouse does not have renal phosphate wasting. It has osteomalacia, and it has huge amounts of osteopontin. That's from Lansky's lab. So this is a complicated drawing I made with Catherine with FEX at the center of the universe, and as a matrix biology and osteopontin processing, et cetera, red and green lights. And then, of course, there's systemic circulating endocrinology biology. But I like to think of it this way. Simplify it down a little bit, and having these nice vertebrate family of proteins regulating and appreciating mineralization. And even their small molecule cousins over here. I didn't talk about polyphosphate. Okay, so I just have 3 slides about the forgotten human mineral, calcium carbonate, because I knew you guys would appreciate that. Probably more work on the carbonates than the phosphate minerals. And this is not published, so this is right off the press. So just 3 slides and I'm done. So the calcium carbonate story in the autoconia. You all, even fish have otoliths, you all have these autoconia sitting in a gel resting on sensory hair cells. And here are the black autoconia. They look like this by SEM in a mouse or a human, it doesn't matter, sitting on sensory nerve cells, okay? And they're in the vestibular apparatus of our inner ears. They sense linear acceleration, gravity. They degrade with time, okay, right? When you're very old, getting there, you start to lose your balance. Elderly people fall. These get dislodged and dissolve, and old people lose their balance. And it's serious. If you lose your balance, you're an old person. Your bones are weak, and you fall and you break your hip, and you get thrombi and embolisms, and it's just a really serious thing. And a lot of it may occur because you've got something wrong with your autoconia, and you're losing your balance. Interestingly, human autoconia normally look like this, but there are case reports that we dug up of autoconia having this shape. And those in the calcium carbonate field know that this maybe looks a bit like vaterite calcium carbonate. And then there's these discoid structures with platelets of mineral. It's also vaterite. This article characterized these minerals as vaterite. But this is not what it's supposed to be. It's supposed to be like this. And you know that calcite polymorphs are more stable, but you always get a bit of vaterite. And organics can stabilize and keep your vaterite. All right? So this is the unpublished work that I'm sharing with you. We decided to see if we could drive calcium carbonate formation into chirality using chiral acidic amino acids. So we used 2 amino acids that are prominent in autoconia, osteopontin, for example, acidic amino acids, the ones I've been talking with. We added the L enantiomer chiral and the D enantiomer chiral. And if you look closely, we drove Away from the calcite polymorph, we drove to vaterite. Not only did we drive to vaterite, but the L enantiomers gave a counterclockwise right-handed chirality, and the D enantiomers made crystal platelets of vaterite with a clockwise left-handed chirality. ASP gave these very nice discoids. Glue gave similar, same chirality issues, but 6 hexagonal subunits in this kind of toroid formation. A racemic mixture, 50-50 of the 2 amino acids, gives you a more typical achiral vaterite disc. We looked at the evolution of growth of these discs and toroids. You first get an amorphous calcium carbonate disc made of all these nanoparticles here by SEM AFM. The platelets, for some reason, start to form at the edge. We're trying to work it out. This is ASP and this is glue on this side. The platelets enlarge, they grow on top of each other, they encroach upon the central core, and in the case of ASP, they fill it right up to the middle. All nanoparticle organization and roughness, and here with the glute. So chiral amino acids can induce chiral biomineral structures. And we're very excited about that. So in summary, this is what I've gone through today using cell culture and mouse model and human data. A story of regulating mineral in skeletons and teeth and autoconia and kidney stones and blood vessels with protein and pyrophosphate interactions. I talked about this enzyme. I talked about the sibling proteins with an emphasis on particular motifs that are important, that accumulate in patients that we can model in simulations and we can test in vitro. I've given you examples of 2 osteomalacia diseases where we know a bit about the enzymes and the substrates. And these rare diseases, these monogenic diseases, are excellent for this because they're not complex diseases. You know the gene, you know where the mutations are, you can make a mouse model, and you can start to figure things out. So rare diseases are really incredible opportunity for us to understand about gene and protein function. And because I was tasked to do that, I inserted a little bit about many tissues were mineralized, but I chose vascular calcification and kidney stones to talk to you about. And I showed you some of our very most recent data that we're very excited about. So thank you very much. These are the members of my group who more or less participated in this way, in this work. I think you saw that I tried to acknowledge my collaborators in each and every slide, so there are many, and they are there. The patients, of course, have been great and helpful, and the families, and the funding agencies. Thank you very much, Karine.